Hello, everyone. This is Ms. Torres again, and welcome to the next presentation. This is Chapter 13, Section 3. It's entitled Cell Transformation. It's going to go on pages, Cornell notes on pages 62 to 63. The date on those pages should either be February 12th or 13th, depending on whether you're 135 or 246. Okay, so according to your period, you should label the page number. In addition, you may want to add the time that you start these notes so you have a good understanding of how long it takes you to actually get through this section of notes. And I can get a better idea of how long it's taking you guys to take certain sections of notes. Okay, so here we go. Today we're going to learn about how cells, what technology we use now in order to do whatever we want to with DNA. Basically, by learning to manipulate DNA, we now are able to do incredible things. And this is just the beginning of what we're looking at today. So today we're starting with the EQ. The essential question of today is, how do scientists study and work with specific genes? So go ahead and write that down at the top of your page. So we begin this section that's of talking about what is transformation. Transformation obviously is not a word that is unique to this process or DNA manipulation. Transformation we use in daily language. To transform something is to change something from one thing to another. But this type of transformation basically entails taking one cell's DNA and putting it inside another cell. So basically transforming the other organism with something other's DNA. So getting the growth gene from, let's say, a cow and putting it into a tomato to get the tomato to grow. That is what transformation is. And how does this occur? Well, first, external DNA becomes a component of the cell's DNA chain, DNA. So the DNA that was taken from, let's say, the cow is put into a tomato and it becomes part of the tomato's DNA. This changes the tomato's DNA, so it changes the cell's DNA. This manipulates DNA to produce medicine, insecticide resistance, in plants, and growth in plants that we've never seen before, like those steak tomatoes, those giant tomatoes that we see. So we basically produce organisms and plants and insects and bacteria that produces medicine for us this way. It basically, if we know it's successful or we know it worked when the DNA is actually integrated into the chromosomes of the thing we want to change. If it doesn't get integrated, it didn't work. So we know it didn't work, but we know it's successful when it happens, when we actually see the tomato growing like really big, when we actually see the bacteria producing, let's say, an insulin protein. So when we see this happening, um, we know that it's happened. That's how we know it's a successful. And there's three types of transformation. We transform bacteria, we transform plant cells, and we transform animal cells. So we have successfully been able to transform three different types of bacteria of organisms using this method. Okay, so this is basically what we do. We basically find a foreign DNA at the top of the page. You see the foreign DNA. The first thing we have to do is that we have to find the gene that we want. So we basically have to sequence the DNA. And right before we took these notes in class, we should have done a lab or basically an online activity, DNA fingerprinting, which basically is how we find the DNA sequences that we want. So we find the gene that we want. Then we use these things called restriction enzymes that we found in being used by bacteria. We actually stole them from the bacteria, and now we have these things called restriction enzymes that actually cut apart DNA. But we actually we've stylized and we target them to basically cut out the gene that we want. So there you have the gene being cut out, and so that's called cleavage. So click on the two orange target sites right there. There, you cut the gene that you want. That's right in between there in the DNA. You get that out. You cut it out of the DNA. You get rid of the blue DNA there, and you put the piece that you want into what we call a vector DNA. The vector DNA, that's basically in the bacteria. the bacterial DNA. They generally have circular pieces of DNA called plasmids. So these circles, we cut one a little slit in them so it opens up the vector DNA or the plasmid, opens it up. We insert our DNA, and there you see the little tiny red one with a little bit of blue. We basically get it to reattach, and now our DNA is integrated into that circle. And then we put that plasmid that now it has foreign DNA in it into the bacteria. And then we let the bacteria just kind of sit there and take it in and use it. And what happens is that the bacteria actually starts copying it. And we make the bacteria really happy. We give it lots of food. We keep it warm in a nice incubator, and we let it copy and copy and copy and copy. And once we've got that copied, then the cells will start to divide and make a big colony of those bacteria that now have our foreign DNA, and then we collect all those bacteria, and now the bacteria start making whatever protein we want. So this is basically what we discovered, and why people with diabetes now are able to live normal lives, or several relatively normal lives, is because we now are able to produce lots of proteins that humans need to stay alive that maybe you don't have. So if you don't your body doesn't produce insulin, we now can grow insulin outside the body, put it in little vials, and then inject then you can use it and put it into your body if your body doesn't make the right kind or not enough or doesn't make any. So this is basically how we produce those things. Okay, so step by step, you cut and remove the human gene of interest with a restriction enzyme. So they're basically little scissors, like I was talking about. Open up the plasmid, the small circular piece of DNA found in some bacteria, with the same restriction enzymes, but it only has to cut once. If it cuts more than once, you tear apart the plasmid and it doesn't work. You insert the human gene into the plasmid, then you put recombinant DNA, a new combination of genes, back into the bacteria, like we just saw in the last picture, and then the bacteria cell contains the gene replications or copies over and over and over again, and you get this colony of bacteria that basically is making a protein that you want it to make. Okay, so the biggest find for us, other than finding out how to sequence genes, was actually finding just restriction enzymes, those little scissors. And here it is, the human gene, the red little line up there, that's the DNA, human DNA. And we cut out the gene that we want. The bacteria, there in the green, has that round blue plasmid inside. It's a round piece of DNA. So we take that round piece out, and we basically cut that with the same restriction enzyme uh, in only one place. We can't cut it in two places. It can only be cut in one place. We cut it open. We insert our desired piece. There we've got it turns orange now that it's been added to the blue plasmid, and then we reinsert this recombinant DNA back into the bacteria with the new gene that we wanted to have, and that's basically a transformed bacteria. Now, if that bacteria starts producing the protein that we want, that's great. We've just successfully transformed that bacteria. So, transforming animal cells. Now, it's much more complicated to do that. So, basically, what has to be done to transform animal cells is, first of all, it has to be done in the egg. Many egg cells are large enough for DNA that can be directly injected into the nucleus. So, animal cells that have enough are large enough so the new DNA can be put right into the nucleus, but it has to be in the egg. It cannot be an already dividing cell. So it eliminates or removes a certain gene and replace it with something else. This is a way to treat genetic disorders caused by a single gene, but there is a risk of replacing a mutated gene by a normal gene. So there is a problem with that. They disrupt the sequence, insert too many copies by accident. That can be a big problem. So animals, pretty much, the only animals that have been done, we've done it on pigs, we've done it on mice, we've done it on rabbits. Uh, Animals we pretty because many of them die in the process. Like if you get like a pregnant bunny that's going to produce like a litter of bunnies, many of them are dead when they're born, and only a few of the bunnies actually get the mutate the upgrade, the different DNA. Um, and that happens. pigs the same way. Many of them die, but a few of them are actually born with the new gene. How do they know that? They generally, what they've been trying to do, just to see if they could do it, they started off doing inserting DNA of a this sea jelly that basically produces it's bioluminescent. It actually glows in the dark, and so they got the gene for glow in the dark, and they inserted it into the eggs, and then they impregnated the female, and then some of her pigs wound up being bioluminescent too. And then they did it to bunnies. They got bioluminescent bunnies and bioluminescent mice. We are able to do that to humans, but it's considered very, very cruel. So people kind of don't do that to human eggs. Okay, so here basically is another illustration. You've got the normal gene, and you cut out the mutated gene. So we're able to fix people like this, and then we put in the modified DNA in the host. So one of the things that this does help us do is fix problems that we catch before anyone is born and fix the genes up. This is still in the very early stages of development in humans, which is why we don't go in and change humans. But we've done it with animals, but it still requires so much. It's still very problematic, and many of the animals wind up with worse defects. So we're not doing it on humans. But hopefully, the studies will continue and we'll get it perfected, and someday we'll be able to fix the DNA in humans. But there is inherent problems. We're going to see a movie where if you start messing with the genes too much and change out and things in, there's a lot of ethical issues with that too that we will discuss further. Okay, so this is basically end of today's presentation, and I hope that you guys do well on this quiz, and you answered all the questions appropriately. And we will make sure you Cornell these notes, now annotate them, and good luck. Okay, if you have any questions, please make sure you bring them up in class, write them in your notes, and ask them in class during the presentation.